First things first, we've woken this morning at VicNoTill headquarters, unfortunately to learn that our special guests, Derek and Tannis Axton, who we were greatly looking forward to hearing from them this morning, are actually unavailable to join us, which is a real shame. So circumstances at their farm today don't allow them to join us this morning. So that's a shame. That's some bad news, but don't despair. We've got some very good news. We've still got our good friend, soil health educator Joel Williams with us, and he's been very adaptable this morning and changed plans a little bit, and we've got some very, very interesting topics to talk about this morning still. So my name is Matt McKinley. I'm a board member of VicNoTill currently. I'll be hosting the webinar this morning. Along with joining us is Brendan Pattison from Mara, a fellow board member. Good morning, Brendan.
Morning, Matt. How are you?
Pretty good, thanks. Pretty good. So we're pretty excited about some topics we've got coming to talk about this morning, Brendan. So Brendan will be with us to facilitate some questions and some discussion. So we've got Joel Williams with us this morning. Good morning, Joel.
Good morning, both. Pleasure to be here. I'm still here in Thursday afternoon, so I'm a bit behind you, of course.
Good afternoon, we should have said. Yeah, yep. So Joel's coming to us from his home in Canada. Is that right?
Yep, that's correct. Yep.
Yeah, good. And we're very, very envious that it's you're enjoying a nice summer over there in Canada as opposed to our winter.
Yeah, that's right. Yeah.
Now, in changing with the topic this morning, we had a quick discussion. We thought on the eastern states of Australia, the seasonal conditions, I think, generally are very, very favourable at the moment with rainfall and temperature, mild conditions, and crop growth is very high. Tends to turn our attention, particularly in cropping programs and pasture programs, to nitrogen management, and it's pretty hard to miss the amount of paddocks with urea spreaders operating in them over the last few days, frantically trying to get urea out ahead of forecast rain, which is a good thing. It's a very positive thing. We've got that yield potential this year to try and protect. So we thought it would be very, very timely, since we have somewhat of a world expert in nitrogen management and foliar nutrition, to change the discussion to that. So nitrogen management, alternative nitrogen management, with a focus on foliar nitrogen management. Does that sound right, Joel?
Yep, spot on.
Yeah, yep. So looking forward to that. In case you're not totally familiar about VicNoTill, VicNoTill is actually a very unique association, a not-for-profit association. So it brings together a very large network of like-minded farmers, and we come together to learn and share information about building better farming systems and innovations. And our slogan there, "Farmers Helping Farmers," I think that pretty much says everything about VicNoTill and what it's about. And this morning's just another example of that, farmers helping farmers. So we would like the webinar to be as interactive as possible. We do invite questions, please. If you have questions, I'm sure there'll be many. It's a very interesting topic. So just if you have a question, would really like to hear it. We'll do our best to put the question to Joel or Brendan or myself. It should be noted, Brendan and myself, we are farmers in the Riverina here, grain farmers, and we do have not extensive, but some experience with using foliar nutrition, but particular foliar nitrogen in our cropping programs over the last few years. So we'll be able to help with some answers to some questions along with Joel. So please use the Q&A feature. So we'll move on to Joel. We'll get straight into it. So Joel, by his own description, is a soil health educator. He's a great friend of VicNoTill. He's a very recognisable face and name right around the world these days, which is a great credit to Joel. So we'll hand over to you, Joel.
Lovely. Thank you very much.
Thank you for your adaptability this morning.
Yes, my pleasure. Penny, I'm just trying to share my screen and it's telling me that the host has disabled screen share. I don't know if there's something you're in you might need to press. I can't share my screen. So yeah, pleasure to be here and to be with you guys. And yeah, we've had so just for any of those last minute people that have just come into the room, unfortunately the Axtons have had just a last minute few dramas on the farm, and they've had to just they're unavailable to come and do the session today. So at the last minute, we've just changed the topic here, and I'll just be doing a lecture on nitrogen there. So, okay, Penny sorted that out for me. I'm just going to share my screen now, and we shall indeed get right into it. Okay, is that working for you guys? Can you see that?
That's good, Joel. Yep.
Okay.
See that cover page. Yep.
Yep. Perfect. Okay, so the session we're going to do is around nitrogen management with a focus on foliar. So I'll start by covering a little bit around generally nitrogen metabolism, nitrogen utilization in the plant, and then we'll move on to discuss a few things around foliar applications more specifically. Yeah, very happy to throw your questions in and we'll have a big discussion there at the end. Okay, so let's kick off. Hold on, there we go. So nitrogen, of course, is a big topic and one of the, as we well know, very important nutrient driving production of our agricultural ecosystems. But on the other side of that coin, nitrogen is also notoriously reactive and a volatile input. And consequently, here we're looking at a map of the world, looking at nitrate leaching. We have a lot of problems with nitrogen use efficiencies. Yes, it's an important mineral. Yes, we've got to manage it for productivity, but it brings a lot of off-site effects, some negative effects. And so that very reactive nitrogen can be converted to things like nitrates, which are highly leachable. And so you can see this is very much a global problem here. Certain hotspots of nitrate leaching, particularly in Western Europe there, many parts there of North America, and certainly over in Asia as well. And so there's a big discussion around alternative strategies for nitrogen management. Particularly specifically for reasons exactly like this. There's a lot of offside negative effects from that nitrogen utilization. And when we look at our nitrogen fertilizers across the board, we kind of get this average figure here of we see around about 50% nitrogen use efficiency. So 50% of what we apply is taken up by the plant. And of course, therefore, the other 50% is ultimately lost from the system. Now, it may be genuinely lost from the system, leached away, volatilized off into the atmosphere, or sometimes it's not necessarily totally lost. It's immobilized by microbes in the soil or utilized by them, etc. So now, yeah, there's been a lot of work on nitrogen use efficiencies, and this figure of 50% is very well validated, but of course it's a range. There's a spectrum. It can be a lot lower than that. It can be a lot higher than that. It really kind of depends on a whole many range of variables and soil types and climatic conditions. But across all of that, we can fairly safely say that there's a lot of room for improvement. If 50% is our general efficiencies, there's a lot of room for improvement there to optimize our nitrogen use. So part of that then discussion is how we then think about nitrogen management from an input perspective, a nitrogen fertilizer perspective. And we have a very dominant narrative in agriculture in terms of managing ammonium and nitrate. This big discussion around these two being the predominant sources of nitrogen in which plants utilize, in which they take up. And if you punch in nitrogen cycle into Google Images, you'll come up with hundreds of them, images, all of them highlighting, nitrate and ammonium are the two forms that the plants take up. In reality, that's part of the picture. It's half of the picture. Plants indeed do also take up organic sources of nitrogen, things like amino acids, even small proteins called peptides. They do take up these organic forms of nitrogen as well. And indeed, these organic forms of nitrogen can be a very important contributor to plant health, particularly, but also plant production. So I think there's a really important point just to raise there is just that we really should be widening that lens a little on nitrogen management. It goes much beyond our inorganic sources of nitrogen. There's a whole other an aspect to this discussion. And so the point is then to say that these different forms of nitrogen that the plant can take up do indeed have very different impacts on plant growth, on plant development, on the efficiencies of the utilization of those forms of nitrogen. So let's just tease out just some of these examples here, just looking at some of these differences. And the first thing to say is that when we look at compare things like ammonium and nitrate, when we look at ammonium, certainly urea is also the same, and some of our organic sources of nitrogen, they are more converted and utilized or metabolized down in the roots. And consequently, by default, they generally encourage more root development. So they stimulate and encourage more root biomass when we use more ammonium forms or that urea form. So we're stimulating and encouraging more below ground biomass. Now, that comes at a real advantage, because early on, if we stimulate and encourage more below ground biomass, more rooting, well, of course, that's setting the plant up ready for the rest of the season. Growing those roots first, prioritizing those roots, particularly in a dryland environment where those enlarged root systems help us access more moisture, then we see those plants, they catch up in the later stages. So they'll prioritize root growth, and then that bigger root biomass will help them catch up and prioritize their above ground biomass thereafter, later on through the season. Now, if we compare that to, say, nitrate, nitrates, when they're taken up, are sent up to the leaf and converted in the leaf. So they're converted above ground. And therefore, by default, they then generally also stimulate more above ground biomass, more shoots, leaf production, etc. And we can kind of see this when we look at, say, for example, cereals. If we look at an ammonium dominant cereal versus a nitrate dominant cereal, it's more lanky, a bit more leggy. It is more top growth, whereas the ammonium dominant cereal, it's a bit more squat, it's a bit more sturdy. It's kind of growing from the base of the plant, not just extending from the tips of the plant, so to speak. So the two minerals, they induce very different phenological kind of habits and growth patterns and habits in the plant species. So that's kind of one first thing there then to point out. And the thing then is to say that the plant doesn't really want ammonium, and it doesn't really want nitrate. It wants amino acids, it wants proteins. So the plant takes those nutrients up. They're really just building blocks. And then the plant has to convert those ionic forms of nitrogen into the more favorable and functional forms that they require. Again, amino acids or proteins, for example. So here we're looking at that conversion process. And straight away, one of the other things we immediately see is that when the plant takes up ammonium, it's a fairly short process to convert that ammonium into an amino acid. And there are some important minerals that the plant requires for this process. It's manganese and magnesium, for example. These two minerals are involved in some enzymes that help to facilitate this conversion. But point is, you can see that this conversion process is a fairly short step to take that ammonium and turn that into the higher quality amino acid and onwards into proteins. Now, when we compare that to nitrate, on the other side, you can see that the pathway of nitrate conversion is much longer. So the plant will take up nitrate, then it has to convert that to nitrite, then that nitrate gets converted to ammonium, and then that ammonium again becomes the amino acid. And once again, there are certain minerals that are important for that process. Oops, sorry, there we go. So the molybdenum being one of them. We need molybdenum for the nitrate reductase enzyme, and we need sulfur to help build that enzyme. So molybdenum becomes really important in that conversion process. Then later on, we need iron to convert nitrite over to ammonium. And then once again, we need that manganese or magnesium, again, to convert the ammonium. So looking at this then straight off the bat, you can see that when we are have a dominant nitrate pathway, feeding our plants a predominance of the nitrate form, it's a longer process to ultimately convert that into amino acids. And that means it's more energetically expensive for the plant. It's more metabolically expensive. Essentially, we're saying that the plant has to use up more energy, waste more energy, expend more energy in converting that nitrate than it does the ammonium. So straight off the bat, an ammonium has a bit of an advantage there from an energetic point of view. And this is now a study we'll share that is then teasing out just this nuance of this question of what is the energy drawdown, the energy demand of the plant in converting different forms of nitrogen. And here we're looking at a study called the carbon bonus of organic nitrogen enhancing nitrogen use efficiency. I'll just read these couple of points out to you. The chemical form of nitrogen taken up, whether inorganic, such as nitrates, or organic, such as amino acids, may significantly influence plant shoot and root growth and therefore nitrogen use efficiency. The key point is that the carbon cost of assimilating the organic forms of nitrogen and turning them into the proteins, that is lower. There's a lower energetic cost than when converting the inorganic forms of nitrogen. And that's mainly because of its carbon content. This carbon bonus makes it more beneficial for plants to take up organic versus inorganic nitrogen. Now, I've got a table that's just going to summarize this study quite nicely, just to reiterate this point here. So here we're looking at 4 different sources of nitrogen: nitrate versus ammonium versus a couple of amino acids. And we're looking at the carbon cost. Again, that energetic cost, that metabolic cost, the carbon cost to convert those sources of nitrogen ultimately into proteins, etc. And so the first thing to look at here is let's compare nitrate and ammonium. And as per the previous slide that I was just highlighting, those 2 pathways, you can see that nitrate requires a greater amount of carbon from the plant to convert that nitrogen. It's more, again, more expensive to convert that carbon into those organic sources. So immediately, this is part of the reason you can see nitrate is less efficient than ammonium, because it's creating a greater drag, a drawdown on carbon. Where does that carbon come from? You know, again, the plant doesn't want nitrate, it wants amino acids, it wants proteins. Where does the carbon come from? It comes from photosynthesis. The plant has to take in carbon, stitch that carbon into the nitrogen to then build those organic sources. So there's a carbon cost, and you can see that the carbon cost of nitrate conversion is greater than the carbon cost of ammonium. Now, if we look at some amino acids here, now moving and looking at those organic fractions of nitrogen, you can see that, again, there is a carbon cost. There's an energy cost in metabolizing and utilizing those organic sources of nitrogen. However, amino acids, being the fact that they are organic, meaning they contain carbon already, means that they come with a carbon bonus. We're getting a rebate on carbon. So, you know, there's not that carbon cost, because unlike these ionic forms of nitrogen, where the plant has to then use carbon from photosynthesis to build those organic forms, we already have some carbon embedded into that amino acid. So there's a rebate, a free carbon bonus. And therefore, when we look at the overall carbon cost of ionic versus organic sources, you can see that because of that rebate, the organic sources then are ultimately more efficient, because, and the key reason is that they've got that free carbon embedded within them. So, and it just makes logical sense. What is the plant doing when it grows? What does the plant want? What's the end point? The end point is proteins, is amino acids and proteins. So if we feed the plant amino acids or proteins, well, then the plant can immediately take those in and more efficiently ultimately convert those into functional nitrogen. So there's some real efficiency gains to be had when we open up the conversation and move away just from looking at nitrate and ammonium and broaden that lens into looking at some other sources of nitrogen. And so here's, again, a little example just to illustrate. We can do a tissue analysis to measure the total nitrogen. And I'm just kind of illustrating a little example here. Here, again, theoretical example, we're looking at 2 different plants. On the left-hand side, you can see that this plant has a lot of that nitrate form of nitrogen, and it has a little bit of nitrogen in the organic form, just for example, a nitrogen containing embedded within an amino acid, for example. So over here on that left-hand side, you can see you have a dominance of nitrates, but just a little bit of that organic source. Now, over on this other side, let's pretend another plant here over on the right. Well, you can see that they have a dominance of, this plant has a dominance of organic nitrogen, lots of amino acid-based nitrogen, and only a little bit of the nitrate. So clearly this plant over in the right has got the molybdenum, has got the sulfur. It's being able to convert those nitrates into the amino acids. So it's obviously functioning at that higher, better, more efficient metabolic level, more plant health, converting the building block. Remember, the nitrate or ammonium is just the building block. We really want the organic sources, and this plant is clearly different than the other and fulfilling that end goal. Now, if we were to send these two plants off to the lab and do a tissue analysis, a wet analysis, where we would digest those samples, we would use something, the lab will use something called a mass spectrometer to analyze these two samples. And a mass spectrometer is going to look at the sample, and as the name suggests, it's looking at all the various elements in there, nitrogen, potassium, whatever, zinc, copper, you name it. And each and every element has its own unique atomic mass. Okay, atomic weight. So the machine will look at the sample and look at all of those different atomic weights, those masses of those various elements, and be able to determine which one's copper, which one's nitrogen, which one's zinc, etc., etc. And then it'll spit out the result. The point is, this is to say that when a mass spectrometer is looking at these two samples, it's looking for that nitrogen, and it's going to determine that nitrogen according to the atomic weight of nitrogen. And when the machine looks at these two samples, this is essentially what it will see. It's looking for that nitrogen, and that's what it sees. It's identified the nitrogen within those plant samples, not if it was in the nitrate form, not if it was in the amino acid or organic form. So you can see these two plants have exactly the same amount of total nitrogen in them. Five little atoms of nitrogen in my hypothetical example here. So the mass spectrometer looks at these and says nitrogen is exactly the same in these two samples. Now, it is, in that sense of the total nitrogen is, however, the form of that nitrogen, the quality of that nitrogen is very, very different. One is loaded with nitrates, the building blocks. One is loaded with amino acids. So I can assure you these two plants would be very different in terms of their overall plant health. We know generally nitrates are often linked to weaker spindly growth, more attraction to pests and disease, for example. So, you know, there's a real problem with measuring your total nitrogen. Don't solely rely on that figure. It's not telling you about the quality of the nitrogen within. And this is where something like a SAP analysis can be useful, because a SAP analysis can tell you what fraction is in the nitrate, what fraction is in the ammonium, and then that total. And of course, the goal is you want the nitrates and the ammoniums to be as low as possible, and the overall organic forms, the total, to be high, but those building blocks to be low, meaning you've got all of those other element cofactors as part of those enzymes to convert that over. So I encourage people to explore a bit of SAP analysis if they're not doing that already, just to tease out a little more of this nuance in the quality of that nitrogen. So what about nitrogen then as a foliar? Strategies to kind of use nitrogen a slightly different way. Of course, the problem with soil applied nitrogen is that the big obvious one is leaching, as we highlighted earlier on. When we apply nitrogen to the soil, it can be converted into nitrates. Doesn't matter if you apply urea, ammonium, or ammonium, they both can be converted into nitrates, which are then highly leachable. And of course, ammonia can also be volatilized off. If we're using urea, it can be converted to ammonia, which can be volatilized off. So there are a lot of issues that associated with soil applied nitrogen. So another tool in the toolbox is to use foliar applied nitrogen. Indeed, plants can absorb nitrogen through the foliar pathway. And they do have very specific means to do that. And urea emerges as a very useful tool in the toolbox for foliar nitrogen. One, because it's high analysis, 46% nitrogen, that means small amounts go a long way. But the urea molecule is also very unique. It has a neutral charge, and that basically means that it's absorbed more rapidly, more effectively than, say, other the inorganic forms of nitrogen, the nitrate or the ammonium. So the plants can take up the urea molecule. It's a very efficient molecule to be absorbed. It's known to be very rapidly taken up, and that's due to the neutral charge of this material. Plus, it has a high analysis. Now, urea is very rapidly taken up. There's only one other form of nitrogen that's taken up faster than urea, and that is amino acids. So those amino acids being smaller, they are very rapidly taken up through the plant. So again, there's an opportunity to foliar deliver amino acids to the plant in that amino acid form, which is again very efficient to be energy saving, energy efficient to be converted and utilized. Then urea is the next fastest after those amino acids, then ammonium, then nitrate. So there I'm referring to the speed of uptake, the speed of absorption. So again, if we can lean towards amino acid based or certainly urea, we have a speed of absorption efficiency gain. And of course, the quicker that you can absorb that nitrogen into the leaf, the less time it's sitting around on the leaf, where it could potentially be washed away, where, okay, urea can still be volatilized off the leaf, of course, as well. But the quicker we get it into the plant, then we're going to avoid that issue. Okay, so urea emerges as a very good candidate for these reasons. And it's also a very good synergist in itself. There's some good studies highlighting that when we put a little bit of urea with foliar sprays of trace elements, particularly this has been shown with zinc and iron, that actually a little bit of urea mixed in with that zinc or with that iron improves the uptake of those trace elements. So urea can also act as a synergist for trace element applications. And again, that's to do with the urea neutral charge. It will bind to the zinc, bind to those trace elements and improve the uptake, the speed of uptake of those traces. So it has a lot of benefits to it. Now, there's a huge amount of work that's been done on urea as a foliar, oodles of studies all around the world. Many, like all studies, many mixed results. Some sometimes not necessarily always favorable, others showing benefit. So there's a really broad array, and of course that comes down to a whole range of different variables: climatic conditions, soil types, soil fertilities, different plant species and varieties. I mean, a whole lot of factors are at play here. But if I just kind of summarize some of those studies down into this slide, there's been favorable results have been shown from urea applications in the range of 10 kgs per hectare of product, 20 kgs per hectare of product, and believe it or not, a study in the UK even going as high as 80 kilograms of product per hectare of that urea, showing improvements to protein and yield. Other studies that perhaps have looked at more percent urea solutions, there's been some favorable results of 3, 4, 5, and 7%, particularly around that 4, 5 kind of percent there. So if I were to convert those percentages to kgs per hectare, well, if any of the listeners are applying, let's say on, for example, 100 liters per hectare of water, that would equate to about 4 to 5 kgs of urea product per 100 liters of water. That's a very small amount. Now, from my experience of dealing with farmers around the world, generally I would say they seem to be applying around that 10 to 15, 20 kgs per hectare of product of urea. It seems to be generally an average application that many farmers use. So it's in that kind of ballpark that practically I see most people using that. Now, the spray pH is also a factor. There was a study there that showed that around the 6 to 6.5 is the ideal overall spray pH for optimum urea uptake. So if you're using, if you have very alkaline waters, if you don't bring that pH down to around that 6 mark, which you should be doing with all your foliar sprays anyway, you will not get as good absorption. And the other point then about urea is that it is carbon-based. Urea has a carbon atom in its structure. So this links to the discussion earlier about amino acids. The reason amino acids are particularly efficient is that we got that bonus carbon, that carbon rebate. Well, we also get that with urea. We've got some free carbon embedded within that molecule, which can then be utilized by the plant, minimizing drawdown on carbon requirements from photosynthesis. Okay, so a lot of benefits there to urea, and in terms of the timings, the studies show if the goal is protein gain, definitely a urea foliar around about that anthesis, right on flowering and the kind of the couple of weeks that follow on cereals. That's your critical window there for protein gains. If you're looking for perhaps more yield gain, the foliar urea has to be a bit earlier, really up until kind of flag leaf. On flag leaf or just ahead of flag leaf is kind of an ideal timing to get that more yield response out of that foliar application of urea. Okay, now the thing about urea is that it doesn't work in isolation. It also needs another very important trace element, nickel, to help convert that urea, to process, to metabolize that urea. So nickel is an essential nutrient, and it is part of a special enzyme called the urease enzyme. And as you might guess from the name, urease enzyme is the enzyme that helps to convert urea, liberate it, break it down, this molecule back here, to separate that, to break that apart so that the plant can use that nitrogen. Nickel and urease is the enzyme, and nickel is the mineral cofactor that helps to facilitate that process. So if hypothetically speaking, we had a nickel deficiency, actually the plant then can't produce this urease enzyme, then the plant actually can't convert urea into amino acids and proteins. It's unable to do that, and that means urea builds up. Urea is accumulated in the plant because it can't be converted over to those amino and protein forms. And that's what can lead to scorch or urea toxicity. And they've shown this certainly with on soybeans who do have a slightly higher nickel requirement. They've shown that actually urea scorch was a consequence of a nickel deficiency. Without that nickel, the plant couldn't convert the urea, therefore urea accumulated and induced scorch and damage of the leaf tissues. So nickel is also a factor in nitrogen metabolism, particularly for urea specifically. Now, there was a study looking at nickel sulfate as applications on barley. Now, it was a pot trial, intentionally very low nickel soil. So barley did show to be responsive to those applications of nickel sulfate in that intentional soil. Now, in an Australian context, of course, a lot of our soils are low pH. So under low pH conditions, nickel is quite available. So if you do have quite a low pH, you're probably not going to have a nickel deficiency. It's very unlikely. On a very alkaline soils, however, nickel availability drops right off. So there could be a benefit from a small application of nickel with that urea in very alkaline soils. Or in soils with no history of organic amendments. So if we're using compost or manure, any animal manures or slurries or compost, then you're usually going to have a nickel component in that organic amendment. But if you have alkaline soils, no history of organic amendments, nickel may indeed be a factor. Now, of course, I say that with a word of caution, be careful. Nickel is a heavy metal. We don't want to be applying excessive amounts. I know farmers have been applying nickel sulfate around about 30 to 50 grams per hectare of product, nickel sulfate, in that kind of ballpark, 50 even up 80 odd grams per hectare. So it's similar requirements to molybdenum for the plant in terms of quantity. So we only need tiny, tiny amounts. I say that with a word of caution, be a little careful, don't apply it willy-nilly. But on alkaline soils, it may be beneficial with those foliar urea applications. And of course, if we did have a nickel deficiency, we're not going to see a response out of that urea foliar. You know, I know foliar ureas is a mixed topic. I know many people are very pro and against. Arguably, part of the reason that people may not see as good responses from those foliars could be an indication that they didn't have enough nickel to then convert that urea into functional nitrogen, into amino acids, into proteins. Here's another study, just again, I'm just really illustrating this. I'm going to just skip to the next slide to illustrate the table here, but just looking at the importance of nickel in that conversion of urea into amino acids. And I'll just cut to the next slide here to summarize this. Here we're looking at a range of different crop species you can see here. And we're looking at measuring the urea inside the plants. And so we're looking at with and without nickel. And so if we just kind of scan our eye down these figures, you can see that for all of these various different crops here, you can see that without nickel, urea is accumulating. These numbers are all higher than when we add nickel. When we therefore convert that urea into aminos and proteins, we can see that nickel declines. So we go from 96 down to 4, we go from 65 down to 2.3, 102 down to 3.6, etc., etc. So urea is declining in the plant tissues because it's being converted onwards into amino acids and proteins, etc. So we can see just that nice little example there. And if we come over here to look at the amino acids, you can see that again, we're looking at comparing with and without nickel. You can see that as soon as we add nickel, we again stimulate that conversion. Well, then the amino acid content of the plant goes up. Okay, so let's look at the amino acids here. They go from 105 up to 165. They go from 65 up to 203, from 3 up to 19, etc., etc. So, okay, as urea comes down, amino acids are going up. And that's exactly what we want. We don't want urea in the plant. We want amino acids, we want proteins. Okay? And that's just for a range of crops highlighting their responsiveness to nickel. Now, so of course, it's urease is the same enzyme that we have concerns about in the soil that also helps to convert urea to hydrolyze urea and volatilize it off as ammonia. And it's the same enzyme. Now, this was an interesting study that showed that actually those very same urease inhibitors that we use to prevent that reaction in the soil, actually they can be taken up by the plant. And those urease inhibitors inside the plant, of course, then block that enzyme and therefore can induce urea accumulation. Without that enzyme, the urea can't be converted, so it accumulates. And just summarizing one line here, this work showed that these inhibitors can induce leaf tip necrosis, resulted from an accumulation of toxic amounts of urea in the plants through that inhibition of the urease activity. So those urease inhibitors can enter the plant. They can also block and stop that conversion of urea within the plant tissues. So just kind of then summarizing some of this discussion around nitrogen conversion. This is just a summary. I'm just representing it in more of a simple image for simplicity's sake. But just to summarize, if we're using nitrates, if the plant is trying to convert nitrates, the plant needs molybdenum, it needs sulfur and iron. So if you're applying nitrate nitrogen, UAN, for example, or ammonium nitrate, for example, that nitrate component needs molybdenum, sulfur, and iron to be ultimately utilized and converted. So maybe factoring in some of those additional elements with your nitrate applications. To convert your ammonia or ammonium, again, I touched on this earlier, we need manganese and magnesium. They're the key minerals that will help to convert that process. And lastly, as we've just discussed with urea, we need nickel. So if you're applying, depending on what form of nitrogen you may be applying, you need these nutrient synergists in order to facilitate that conversion. Okay, so put them in your radar, put them on your radar as well. So now let's kind of then think about this through a lens of foliar. Feeding through the foliage has a lot of advantages. It can very much bring a whole host of benefits to crop production. For example, bypassing soil imbalances. Minerals in the soil can accumulate. They can be in high levels. High levels of certain nutrients can antagonize the availability of others. They can lock them up. They can block the availability, block the uptake, or bind to them and antagonize them and lock them up. So there are a whole host of soil imbalances and issues that we can bypass by delivering the minerals directly into the foliage, into the plant. Okay, so we avoid, of course, things like leaching. There's no leaching associated if we don't put the nutrients down into the soil. Less volatilization off the leaf surface. Again, I stress leaves can volatilize off. Urease enzyme is active on the leaf surface, so you do see volatilization off the plant, but it's generally less than what you see from the soil. And less fertilizer required. We're using smaller, more targeted amounts of those nutrients when foliar applied. So of course, we can't be applying 100 kgs, 120 kgs of urea onto the foliage. We're probably going to burn. So we're using much smaller amounts, but we're targeting them directly onto that foliage where they can then be absorbed. However, I know that many of you will probably have mixed feelings about foliar applied nutrients. They're very variable. Sometimes you do see a response, sometimes not. And it's important to understand how to design and how to apply a foliar spray in order to get that good response. There are many variables. There are many factors. I acknowledge that. It's not a guaranteed sign of success just because you foliar. You've got to design it and apply it in the right way. And let's tease out just very quickly a few of those factors. We could do a whole lecture just on this, so I just want to cut to a couple of key points. But first thing is to understand the pathways of uptake. Plants take up nutrients through the foliage, one through the stomata, through those open stomata, and secondly through the cuticle, through that waxy layer, through micropores in that cuticle. So therefore, understanding those two pathways of uptake means firstly that, okay, stomata are more so on the underside of the leaf. Many of those micropores are all over the leaf, but particularly concentrated also on the top, but they're all over. So if we're trying to optimize leaf coverage or optimize or maximize both pathways of absorption, then we want to make sure that our foliar spray reaches the top of the leaf and the underside. Covering both sides of the leaf covers greater surface area for uptake. And then the obvious point is then things like the environment, the humidity. When we have dry air, lower humidity, well, those micropores are closed. When we have more humid air, so early morning, late afternoon, etc., well, then those micropores are open because of the humidity. And it's that extra humidity that then helps the foliar solution be absorbed into that plant. So there's no point in applying foliars in the middle of the day in the heat. Okay, you want to do it early morning or even late at night, late in the afternoon and through the night. More time when those leaves are open, the pores and the stomata are open. It's the same deal. Stomata are closed in the middle of the day. The nutrients are not going to be absorbed through that stomata when those stomata are closed. So, environment, these kind of factors are very, very important. So steer your foliars to early morning and late afternoon, evenings. And again, I've just summarized this. This is a lecture we could do a whole hour of, so I want to cut to the chase here. But there are many variables that influence the success of a foliar spray. You can't just throw it in the tank and spray and pray. You've got to design it for success. And that means considering things like this, the formulation. What is it you're applying? Are the nutrients soluble? The concentration? What are you mixing together? Different nutrients or synergists, some can antagonize. Put some thought into the formulation. What's your water quality? Is it high in bicarbonates? Is it very alkaline? Bring that pH down, for example. Use as clean water as possible. All of those kinds of things like that are very, very important, including the concentration of that mix as well. And then, of course, the obvious things like application considerations, your nozzle size, your droplet size, etc. All of those things are important. The crop plays an influence, the crop characteristics. Some plant species are more responsive to foliar sprays than others. Younger leaf tissues are more responsive than older leaf tissues, which get a bit older and more mature, hardening off. They're a little less, they absorb a little less through the foliage. So, the crop, things like brassicas, of course, they have very waxy leaves. They're generally a little less responsive than, say, other plants that don't have such waxy leaves. So, the nature of the crop also plays a big factor. And then the environment is also a big one. And I just gave an example of that there. With humidity, temperatures, time of day, these kinds of things. So, all of those things, you have to be designing for all four of those considerations in order to optimize the response of that foliar spray. And, okay, in the example of urea, as I mentioned, spray pH, make sure it's 6.5, maybe synergistic nutrients there. Chelators, the use of carbon-based chelators is another very useful aspect of the formulation that's very important. When we wrap up the nutrients with a chelator, complex it up in a carbon source, well, that chelating agent helps with the uptake of those nutrients. So all of those things are variables. There's many points to discuss in each of those, so I'm sorry I just have to brush over those, but just to highlight that, again, don't just mix it in and spray and pray. Think about it, design it accordingly, and that'll improve your chances of success in terms of getting that response. And I just quickly tease out one of those considerations in terms of tank mixing your spray, and that's the importance of using carbon-based inputs or carbon sources in with those nutrients. You don't want to apply nutrients on their own. You want to mix them with carbon compounds that will then bind to and complex those nutrients, for example, in this image that I'm illustrating here. When we complex and chelate and bind those nutrients, well, these chelating agents, they facilitate the absorption. And the reason is that when we apply the nutrient on its own, and this is particularly true of, say, cations, positively charged nutrients, they are quite strongly attracted to the leaf surfaces which are negatively charged. Plant leaf surfaces are negatively charged. So those cations, they can get stuck on the leaf surface. That strong charge attraction creates a bit of a bottleneck. So Consequently, by wrapping up the nutrient with a carbon source, we neutralize that charge, and therefore this facilitates a more rapid uptake. So carbon sources are things like molasses, very commonly used, amino acids, fulvic or humic acids, fish hydrolysates, seaweed extracts, any kind of plant type extracts. All of these things are carbon sources. I know many farmers who use often just molasses with their urea, very happy with that. It works very well. Humic acid would be another very common one that does go very well, fit very well with urea specifically. But any of those carbon compounds will help to bring this benefit. Okay, I'm mindful of the time. I was going to say a few quick things about free nitrogen fixation, but I'm kind of thinking I will just scoot through this because we should open it up to some discussions. That was the core of the discussion around the foliar element. I'm just going to probably cut through some of these slides just so we can wrap up, but just to say that don't forget free nitrogen. There's lots of nitrogen gas in the air, and we want to also try and access that. And this is part of the reason we might consider foliar applied nitrogen. Because, okay, that was just to say that many crops can also fix their own nitrogen. It's not just legumes, but many of our cereals, etc., can associate with other nitrogen fixing bacteria. So that was just to illustrate that. This is the study I wanted to come to. The reason we want to then think about foliar nitrogen is because that will help us use less in the soil. The evidence is very clear that you will get a better response from a foliar nitrogen when you are also reducing your soil applied nitrogen. So cut soil applied nitrogen down and substitute it up into the foliar. And that's when you get a better response from that foliar. If you do full rate of nitrogen on the soil and then try and do a foliar, well, you're probably not going to see a response. Cut nitrogen down, lower your application rate in the soil, and then do a foliar. That's where you'll see a better response from that foliar spray. And so this is what you all know already. I've just put some graphs up here to illustrate this, but if we have too much nitrogen in the soil, of course, that suppresses nitrogen fixation. And that's true of legumes, but it's also true of all of those free-living associative nitrogen-fixing bacteria as well. So, of course, as the soil nitrate, here we're looking at nitrate as it increases, well, you can see that the amount of nitrogen fixation declines. Here we're looking at the available nitrogen to phosphate ratio, and we're looking at the nitrogenase activity. That's the special enzyme that nitrogen-fixing bacteria use to unlock nitrogen gas. You can see that as our available nitrogen increases, that the activity of that enzyme declines. So as you all know already, more nitrogen in the soil typically means less N fixation. So by substituting up to the foliar, you're putting less down into the soil, opening up the opportunity to support and sponsor more of that free nitrogen coming into the plant. That was a study just to illustrate that when we over-apply nitrogen, we see less nitrogen fixation. That was a recent one from last year. Over 4 decades, they showed that nitrogen fixation was dropped by about 50% after 4 decades of nitrogen fertilization. And another study saying exactly the same from Rothamsted in the UK, over 100 years history of nitrogen fertilization. They showed depressed nitrogen fixation capacity in the nitrogen fixing microbes there. I was going to go a bit more detail on those, but I'm just scooting over those to wrap up. And then lastly, just to say that there are also these other important minerals that again are important for nitrogen fixation. You cannot fix free nitrogen from the air without molybdenum, without iron. They're part of this nitrogenase enzyme that helps the plant, helps the microbes, the bacteria, unlock and grab that nitrogen gas. So we need molybdenum to get free nitrogen. We need iron. Again, nickel emerges. It also plays a role in this enzyme, hydrogenase, which also plays a role in nitrogen fixation. And also phosphorus, very important. N fixation is an energy intensive process, and phosphorus helps to bring that energy. So those first 4 there, molybdenum, iron, nickel, and phosphorus, all nitrogen fixation, be it with legumes or free living, all of those N fixating bacteria require those minerals. And if you have a limitation of molybdenum, of iron, nickel, or phos, that will limit your ability to access some of the free nitrogen. And if we then widen the discussion to legumes, on top of those first 4, legumes also have a very important role for calcium requirement, for calcium, boron, cobalt, and copper. They also have a very direct function in nitrogen fixation. So any limitation of any one of those minerals, you will also limit the potential of free nitrogen that you can access. So in summary then, I would just say that I'm not suggesting that, foliar is the most important pathway for nitrogen uptake. Of course, roots play a role. Of course, roots are the dominant source of nutrition uptake for plants. Of course, nitrogen coming in through the roots is important. I look at the foliars as simply to say that foliars present a real opportunity to complement your soil-based management strategies, to complement them, to prove those efficiencies, use a little bit less, be a bit more targeted, but you've got to do it right. Think about the formulation in those kind of considerations, the carbon source, the pH, think about those other factors in order to ensure your chances of success. But I believe that foliars can be a very useful tool as part of an integrated nitrogen management strategy. Okay, and I'll close it there.
Yeah, go, Brendan. Yeah.
No, you're right. No, thanks, Joel. That was a really good insight into a lot of that foliar nitrogen. You've certainly highlighted a lot of really key points in there. Certainly that pH was certainly interesting with the solution. Your nitrate conversions versus ammonium conversions in the leaf was certainly very interesting here. And yeah, I certainly found it very interesting. Did you say something, Matt, there?
Yeah, I just had a question. There's actually quite a number of questions come through, Joel. A lot of questions, so we probably won't get to them all, but just going right back to the start of your presentation with your reference to nitrate. So nitrate being not always or often an undesirable form for the plant to be taking up. Mm-hmm. That said, is there a role for nitrate in the plant, particularly with biomass accumulation and for fodder purposes and so forth?
Absolutely. It's an excess of nitrates that become problematic, or perhaps an imbalance of nitrates versus some of those other forms. I wouldn't say, therefore all nitrate is bad. Some is you want some in the plant. It also helps the plant to maintain its electroneutrality, its balance. So, nitrates is okay. It's just that we typically in production agriculture, because irrespective if you apply urea to the soil or ammonium to the soil, both of those two forms can end up being converted by the bacteria in the soil into those nitrates. So it's very common that we do see an oversupply of nitrates in the plant, irrespective of what form of nitrogen that we use. So a little bit of nitrates, perfectly fine. Yes, of course, it plays a role. It's just generally we see a bit too much of it. And you can compensate that by making sure you push that molybdenum and the iron and the sulfur to help convert those nitrates, as we mentioned. But yeah, a little bit is fine, just not too much.
Very good. There's a good question here, Joel. How can we get our foliars to stick to our plant leaves when there's a lot of moisture about in the morning, like heavy dews? Is there a preferred wetter or sticker we can add to make all that work better?
Yes. So again, actually, some of that morning time dew is and morning time moisture and humidity is also favorable for the uptake of those nutrients. Again, as we mentioned, the high humidity, those pores being more open. So there are some advantages to that. That's an ideal time. But of course, if there's too much heavy dew and it starts dripping off, then sure, you might want to add a wetter sticker. And yeah, I would support that. I think that comes under the formulation considerations. Yes, I would put a carbon source. Yes, I would manipulate the spray pH. And yeah, I would likely put in a wetter sticker. I don't have any particular recommendations on which one. There are many out there. I'm sure some are better than others or whatnot, but I'm sure they all do the job. But yeah, I would, for any foliar spray, I would be inclined to be putting a bit of a wetter sticker on. Yes.
Okay.
Hey, Joel, just to clarify with regard to the nickel and moly and iron and zinc and so forth, the enzymes you spoke about and the role that they play in the solution, what's a good way, like before we make the application, to determine whether that's actually necessary or not? So to determine whether our levels are adequate or not, do we look at quantity in tissue or sap, or how do we go about that?
Yeah, sure. And yep, I agree. I would always recommend testing first, taking a tissue analysis, a plant analysis, and determining your requirements. I don't support applying nutrients for the sake of it. Let's be a bit more targeted. Let's identify what our actual limitations are and then go and address those. Focus and spend our dollars accordingly. Now, yes, you could use either a traditional tissue analysis, that's quite okay, or you could use a SAP analysis. Both have some strengths and weaknesses. I would encourage people to use one or the other rather than none. Definitely, to use at least something. Check what you're actually limiting first before you apply it. That's true of all things. You don't want to be inefficient and apply things for the sake of it. You don't want to induce greater imbalances or induce a toxicity. So you want to, particularly some of those traces, so you want to make sure that you do actually need it before you apply it. So either a tissue or SAP test. And I think the SAP test is particularly has a particular advantage, as I mentioned, just because it then also teases out specifically the nitrate and ammonium component, not just looking at the total nitrogen. So I do lean a little bit towards the SAP analysis for that benefit. Yeah.
Joel, is there an upper limit to EC for our spray solution without getting any leaf burn if we're mixing a few other components with it, like compost tea or something?
Yeah, sure. So generally, they'd say that your overall EC, an ideal EC would be about 1.5 to 3 millisiemens per centimeter. Yep. Is a good kind of concentration. If you're starting to add biology, biofertilizers, microbials, I've heard that perhaps you shouldn't be exceeding 2.5 as your overall EC. So yeah, definitely you want to lower that if you are adding microbials. And I would also say that if you are adding microbials, that would overemphasize the importance of that carbon-based component. Make sure you add the molasses, the humic, the fulvic, the fish, whichever, those carbon compounds will bind to the ionic minerals and therefore help to buffer them, make them not so harsh against the biology. So no greater than say 2.5 if you're adding microbes, and maybe go a little higher on the carbon-based components.
Okay. Yep.
Okay, Joel, here's an interesting one. Questions come in. What do you recommend for determining if there is a deficiency in the microbes needed for fixation?
So, sorry, could you just repeat that again for me?
Okay, recommend determining if there's a deficiency in the microbes needed for fixation. So I'm assuming that's in soil.
Yeah, correct. Yeah, so again, that's a good question. It's a little trickier. Yes, you're right. You might want to be doing more of a soil analysis to determine some of those. When I talk about those mineral constraints to nitrogen fixation, that slide that I put up at the end, that list of minerals, yeah, there I'm referring to those minerals being essential for the microbes, not necessarily for the plant, for the microbes in order to do that job and fix that nitrogen. So that might therefore mean you might want to be considering more of a soil analysis to assess those. Now, that said, I know and Pata, you mentioned this once when I was on your farm. I know that that said, a foliar analysis, tissue or sap analysis, particularly something like molybdenum, you can use that still to diagnose, particularly on a legume, because even a foliar spray of, say, that molybdenum, they will translocate that molybdenum down to the nodules, and where then those nitrogen fixing bacteria will be able to use that. So there is a bit of a link with the sap. And Pato, I remember, if you want to throw in a comment there, I remember you told me once you threw some molly, you saw very poor nodulation on some beans and then applied a molly. Is that right?
Yeah, I noticed there last year it was where we had our beans weren't nodulating as well as they probably should have. And after a sap test, there was, no molly in the plant. So we went forward with a foliar molly, and a couple of weeks, 2 or 3 weeks later, we started to see a lot more nodulation, especially out on all the lateral roots, which was a real good positive, and went forward from there.
Okay, so that's a good example where still a plant and a foliar can overall help, particularly with the legume example. But otherwise, you might want to be looking at your soil analysis to determine. You can test for molybdenum, you can test for those other trace elements in soil analyses to determine if they're potentially limiting or not. Yeah.
On the adding nickel there, Joel, with our acidic soils we have here at home, you were saying how nickel was more available in an acidic soil.
Yes.
Would we still get a benefit by adding a small amount of nickel with our foliar spray if it's very minute in our actual plant?
Certainly, possibly. I guess I would encourage you to do maybe some small trials on that first, because unfortunately we don't have we can't really do a tissue analysis or a sap analysis for nickel. It's such small amounts that it's required, and we don't know ideal levels. We just kind of don't have that information. But I would say there may be other antagonisms. So, many of those other trace elements that are all kind of 2+ charge. So things like copper and zinc and iron. Nickel is the same. They can be competitive with each other. So if you had a soil that was exceptionally high in copper or zinc, for example, or any one of those, it could actually be antagonizing the uptake of that nickel, even if the nickel is there, present, available. You can see those kind of antagonisms. So, and I've seen this also even in the context of a foliar spray. Some of the nut trees, pistachios, no, not pistachios, what's the other nut I'm thinking of here? Walnuts, pecans, that's the one. Pecans actually have quite a high nickel requirement. It's very essential for them. And if you are applying too much, also through foliar sprays, if you're treating some of those other 2+ charged trace elements, they can actually induce a nickel deficiency, and it's a big problem in pecan trees. They have quite a high nickel requirement. So so again, to come back to your question, if some of those other minerals were rather high or excessive, they could be blocking the uptake or availability of that nickel. So it may be possible that even under acidic conditions, that nickel may be limiting. And the amount through your foliar that we're talking about is quite small, 30 grams, 50 grams. I mean, it is a small amount. I guess I would encourage people just to if they're doing a foliar urea, do half the field with straight urea and then throw a bit of nickel into the tank and do the other half and do a few tram lines and see if they get a response to it.
Yeah.
Okay, Joel, probably roll quite a few questions into one here, if that's okay. A lot of questions about the humidity, daytime humidity, summer foliar spraying, winter's foliar spraying, and continuing on from that, even time of day, like in the evening with daylight and darkness, just in relation to whether the stomata's open or closed with daylight. Yeah.
Yes. Okay. Sure. So as I mentioned, the time of day is morning and night are your best ones. Actually, there's been a study that showed that simply the mere presence of light also encourages uptake. So, there's an argument that might say that the morning is a bit better because you've got a bit of light coming around for that uptake. On the other hand, the slight negative of the morning is that then it's going to get hotter, and then that foliar spray is going to start to dry out. And as that foliar spray dries out, if the nutrients haven't been taken up yet, then they can start to crystallize out on the leaf surface. So from that then point of view, perhaps you could argue that the afternoons and the evenings are perhaps a more preferred method, because you'll have a bit more longer moisture and humidity there and wetness there for the solution, the nutrients in that solution to be absorbed before the solution then dries out and the minerals may crystallize. So that would be there's no right or wrong answer to that one, but I think you've got to do what's convenient in your farming system and your time. So, but in terms of then summer and winter, I would say that, of course, you'd have to assume that in the summertime, the plants are going to be a little less responsive, simply because for you guys, it's going to be so much hotter, and they're going to be a little bit more stressed, etc. So again, it would be critically important to target your applications in the mornings and evenings in the summertime. But in the winter time, you've got a little more flexibility. If it is a slightly cloudy day, if it's a little bit of a cooler day, if the stomata are open, fine, you could be foliar spraying in the middle of the day. That's okay. If it's cloudy, if it's a bit cooler, that's quite okay. So you've got a few more opportunities in the winter, which of course works well for your growing season, being winter dominant anyway. So,
Joel, with the scale of some of the properties, if we have to go to a granulated product to get over our country, because we just can't get the foliars out in that optimal period, as you've just discussed, What sort of damage are we going to do to our soil or our bringing up our free nitrogen through microbial activity? Are we going to do a lot of damage?
It really depends on your application rate of the soil applied nitrogen. You know, some amounts are perfectly fine. And that's where things like split applications, you know, trying to stagger that out with more frequent, more smaller doses can be very advantageous. So it's not one big burden up front. You know, I would support that kind of strategy. Yeah, it's not to say that therefore it's just a blanket nitrogen is all bad. I mean, it's going to depend on the soil type, how big or light or heavy it is, its organic matter, how much buffering it's got there. So, you know, there's a lot of variables that would be at play. But I would say that if you can spoon feed and split out those applications, that would be one advantage. And again, I mentioned that principle of carbon-based fertilizers that applies to granular. If you can coat your granules in a carbon, using one of those carbon inputs, things like a humic acid or even a molasses or some of those other carbon sources, doing a carbon coating of the urea granule or the nitrogen granule, then you're putting a little bit of that carbon there that as the granule solubilizes, you'll have some of those highly available carbon forms that can, again, bind to that nitrogen and also stabilize it. So I would say I would overemphasize the importance of carbon-based nitrogen when we are using granular applications, because that carbon will help to stabilize that nitrogen and cause less of those problems that you're concerned about. And I think the point is very valid. You know, we know, of course, granular spinning is much more efficient. Foliars may take a bit more time. I would then argue in that context too, if you are a bit pressed for time, you know, I would be prioritizing your poorer fields for those foliar sprays. You know, prioritize them if time is a real issue. If time is a real issue, I would be prioritizing my poorer fields, you know, in that early morning and in that late afternoon, evening time frames. You know, prioritize your poorer fields that might need a bit more of a boost with those get the variables perfect for those, and sure, maybe you can be a little bit more relaxed on some of the other fields. So, but yeah, of course, I acknowledge spinning out fertilizer is going to be a bit quicker, and I guess I would encourage people to think about trying to integrate the two together. You know, can you go with a slightly lower application of nitrogen onto the soil and try and top up and improve those efficiencies there through the foliar pathway as well? So an integrated approach, I think, is the key message.
Yeah.
Yeah, Joel said, I guess with reference to integrated approach, I guess it may be more of a comment than a question, but I guess for a lot of us with changing systems and changing ideals, that the ideal situation is not to be operating in a low nitrogen situation in the first place, where you need to be chasing your tail and resort to granular applications. So, yeah, can you expand on, you know, most farmers have got a pretty robust focus on diversity and fostering microbial activity in the soil, fostering free nitrogen bacteria and so forth, just to try and avoid that really low nitrogen situation. So, yeah, is there any big standout tips to try and avoid that situation, to try and complement a foliar program and not have to resort back to a sort of a bulk granular program?
Yeah, I guess there's a lot of variables to answer that question in different contexts of farms and how they each manage their time and their workloads and all of those kinds of things. I agree that, of course, you don't want to be chasing your tail. You want to be a bit more proactive, and in terms of you know, being a bit more organized or monitoring that nitrogen, to know that if you've got a limitation coming up, you know, you don't want to discover that at the last minute and then, you know, induce that time management problem. So I would encourage people to be a bit more proactive, maybe do more regular sap or leaf analysis to be checking that nitrogen through the season as the season is evolving. So you can maybe, if you see that it's low, that's the beauty of an analysis. You know, often you can pick up that limitation before it's physically expressing itself. So I guess I would encourage people to invest that money on doing a bit more closer monitoring of the plant level, so you can get ahead of the game. And, you know, there's a real discussion around front loading nitrogen versus spoon feeding. And again, there's no right or wrong to this, and you see all sorts of different contexts where one can be better or the other. I know, for example, in the UK this year, they've had a very dry growing season so far, and so those that were using a little bit more nitrogen up front in early spring, when there was still that little bit of moisture around, that proved to be very beneficial for them, because then the season quite rapidly started to dry out, and then trying to catch up and get nitrogen on later was not quite so effective. Now, had that been a wet year, well, those upfront nitrogens would have been not the best solution. It would have much been better to spoon feed and stagger those out. So, there's no right or wrong to these situations. It gets very context dependent. And I think for me personally, that's one of the reasons that I also really value the foliar approach, is that you can manage the season accordingly. If there's no moisture, if yield potential looks low, well, don't be out there spraying, don't be out there putting that nitrogen in. If you did front load and then it went dry, well, you've put in a lot of capital investment there. If you play the ball game through the season, and you're being a little bit more, proactive and reactive to that season, well, I'd argue there are some real benefits to be gained from waiting and using foliars. If you know the moisture's there, if you know the yield potential is there, then you can justify the expense and the time and the labor to go and do that. So I think that's one of the reasons that I'm also a bit of a fan of the foliar approach.
Yep. I guess it'd be a common theme and a common observation amongst VicNoTill members and people within the VicNoTill network that, as people sort of transition away from a synthetic system, the benefits start to build and gain, and nitrogen after a number of years tends not to be, or the curve sort of flattens. It tends not to be the wild variations in nitrogen availability that we saw in a more synthetic approach to farming. Sure. So I think, as that approach builds, it really opens the door for the smaller, as nitrogen becomes less crucial. It really opens the door for the foliar strategy.
And again, I know I had to brush over those couple of slides there, those studies there at the end, but just highlighting that, the evidence is very clear. When we have less nitrogen in the soil, microbial nitrogen fixers are more active. That nitrogenase, they're producing more of that enzyme. There's quite a few studies that all support this. So, that's one of that other critical benefits, is that as you wean down off that synthetic end, you're opening up the door, or opening up the opportunity to facilitate and foster some more of that natural nitrogen fixation.
Such a common observation and catch cry over the years with high input, like high nitrogen input urea use, that seemingly the more you use, the more you need year on year. Breaking that cycle in the first place is probably a good starting point.
Yes, that's right. And that's what those two studies alluded to. One for 4 decades, one for over 100 years, both showing that with and without excess nitrogen in the system, as soon as you have surplus nitrogen in the system, the microbes get lazy. They don't need to be fixing nitrogen anymore. So we've got to be thinking about dialing it down from the soil, and that'll help to encourage that natural stuff. But for those, let's be honest, production is still important. You've still got to have a production focus as well. So that's where the foliar can then top up and still help make sure you're delivering extra nitrogen, and with that kind of production goal in mind.
Yep. Can you see, Joel, moving forward, that we can get away from higher nitrogen use with a lot more diversity and microbial activity in our soils, like through our foliar programs? Like, can we reduce our total N to a pretty low number?
I mean, when we have greater, again, I would say as a generalization, there's a lot of studies that would support that the more nutrient saturated the soils are, that the microbes, and this is beyond nitrogen, this would be true of many other minerals, but the more nutrient saturated the soils are, then microbial activity can often be compromised. We know with lots of P, mycorrhiza shut down. We know that, again, the example of nitrogen we've already discussed, and fixing bacteria shut down. So yes, I would say that Yeah, I would say that the more we can dial down those inputs, the synthetic nitrogen, the more it opens that door for the biological activity to recover, to be more active. So yes, I think, and again, that point extends beyond nitrogen. It's why I am a supporter of the foliar approach in terms of for all nutrients, not just nitrogen.
Yeah, no, fully get that one. Yeah.
Okay, Joel, there's so many questions here. We're certainly not going to get to them all, and time is moving on, but we might make this the last one before we wrap up, if that's okay. But there's quite a theme in the questioning coming through, just about with sap tests and observations with ammonium to nitrate ratio. Is there actually like an optimum balance there that do you aim to achieve? And if there's an apparent imbalance, what are the reasons it could be causing that? Is there some issue with reductase enzyme imbalance or something there, if there's an imbalance between ammonium and nitrate? Mm-hmm. And what is the parameters around ammonium and nitrate ratio?
Yeah, sure. Again, it's going to depend on different plant species. So different crops are going to have kind of different slight requirements. And I would agree that I would prefer to see ammonium being always higher than nitrate. I think, ammonium is more favorable than that nitrate fraction. But ultimately, going beyond that, as I kind of alluded to at the start of the lecture there, in a way, I'd like to see both ammonium and nitrate both quite low as a percentage of the total nitrogen that's in there. You want more of it in that amino acid, more of it in that protein. So in a way, you could argue that we want them both to be as low as possible. And that gets a little tricky in managing some of our sap analysis in the guidelines, is because, with that production focus, many might be looking at that as low and thinking, well, I need to apply nitrate, I need to apply ammonium if it's low. So it's a little bit of a different, looking at it in a little bit opposite ways. It may not necessarily be that you do. If your total nitrogen is good and in the range, but those are low, I would say that's optimum. If the total is low and nitrate and ammonium are also low, then sure, you need more of those building blocks to then help build and convert and synthesize the organic and organic amino acid and protein forms. So I don't have a specific answer in terms of an exact ratio. I think it's going to be a little different. Different species have different nitrate and ammonium requirements, but as a generalization, I would say I would definitely want to see ammonium to be greater than nitrate. And then I would still want to see them both, relatively speaking, lower as compared to the overall organic forms and total nitrogen as well. But yeah.
Okay, thanks, Joel. Brendan, I think it's about time we wrapped up there. Sorry to questionnaires that we didn't get to. There's a whole bunch of questions there. We just really aren't going to have time to get there. Sorry. So I think we covered a lot of good topics in the questioning and discussion. So thanks, Brendan, for your insightfulness. Very special thank you to you, Joel. Particularly for your last minute adaptability. So very, very good session. To the listeners and participants, it's been a great pleasure on behalf of VicNoTill to bring this to you this morning. Joel Williams, he's a very insightful person, isn't he? And he's always jogging your brain, always making you think about what the next thing is, how we can improve our systems. And that's pretty much what VicNoTill's all about. Our ethos of farmers helping farmers. It's alive and strong. It was alive and strong this morning. If you're not a member of VicNoTill and you'd like to support VicNoTill, VicNoTill is it's a not-for-profit organisation. It's a member-based organisation. The best way to support VicNoTill is, if you choose to, become a member. That's very simple by going to the website and follow the prompts there. If you want to give Penny a call to discuss things, that's fine. So I think we'll leave it at that. A really good webinar. Thanks, Joel. Thanks, Brendan.
Awesome. Thank you very much. And thanks for having me. And yeah, thanks to VicNoTill for putting it together. And hopefully we can get the Axtons back on next time.
Great work. Thanks, guys.